I'm so happy to be here. It's great to see all of you, and thanks for coming out. So today I'm going to talk about coral health from microbes to branches, which is my hope that you're interested in both microbes and the coral animals themselves. Oops, I just gave something away. Before I really get started, though, I just wanted to thank everyone who's been involved in this research. CCMI and University of Florida, as Beth mentioned, have had a collaboration over the past several years, and I've been lucky enough to be a part of it now over the past year and a half. Through this collaboration, we also have wonderful funding sources here in the Caymans as well as in Florida, which are listed here. And I just wanted to thank the collaborators both at CCMI and in Florida, Carrie, Dagny Elise, and the CCMI interns and staff, Tom Fraser, Julie Meyer, and Monica Schul, Lindsay, Larry, and Doug. And we've had some amazing volunteers come out and help us with some of the work we're doing too. And so if you're interested in that, I'm sure the CCMI staff who are here would love to tell you about that too. So as members of the Caymanian community, whether you're a tourist or a visitor or live here, I think we can all agree that coral reefs are very important. They're important because they're ecologically diverse. They're economically important. Probably 1 million visitors come to the Cayman Islands every year just because of the reefs. And the foundation of reefs and why we're all here are because of these corals. And I want to start kind of simply, although this is not really a simple question, but what is a coral? Is a coral an animal? You can raise your hand if you think it's an animal. Cool, awesome. Is a coral a plant? Raise your hand if you think it's a plant. What about a mineral? What about all three? Oh, you guys are great. There's already a great amount of coral knowledge in here. And so here's just an example of one of these beautiful organisms. And you're right, they are all three. They're part animal, they're part mineral, they're part plant. So if we were to cut a coral in half, like this, it's really just that top layer of the coral that's living tissue. The rest of it is the calcium carbonate skeleton, similar to how our skin covers our skeleton. And a coral is really full of interconnected polyps. So if we were to zoom in, we would see all of these little yellow circles here in this case with the tentacles that would reach out to catch microscopic organisms in the water column for the coral to feed heterotrophically. And then if we were to zoom in on one of those polyps, which cartoonily looks like this, inside of the coral tissues are the zooxanthellae. So that's that plant-like part of the organism, right? They photosynthesize, which means they make oxygen and they make sugars to feed the coral by taking in carbon dioxide. And it's this relationship between the coral and the zooxanthellae that I think most people are familiar with, right? Heard of zooxanthellae? All right. And if not, yes, you've heard of them now. Covering the surface of corals is a mucus layer. And I'm sure no one here has touched a coral, but if you have, don't do that, that's bad. But if you've accidentally done it, don't do it by accident in the future, you might be able to tell it's kind of slimy. And that's its mucus layer. And that mucus layer has a rich bacterial community. And that bacterial community is really, one of the things we think it's responsible for is the health of the coral. So it's that first barrier, like our skin is to the environment, between a coral and the environment. So it influences what pathogens that might be able to come in. It influences what pathogens are not able to come in. So it can outcompete potential bacteria. And this microbial community is really rich. It's dense. There are tons and tons of bacterial cells on a coral surface. And if you were to look at that underneath a fluorescent microscope, it kind of looks like a sea of stars, which hopefully you can see in that corner over there. And so when I'm talking about a coral, I'm talking about all of these different aspects of the coral. And if you've gone snorkeling or diving on any of the reefs around the Caymans, you've probably seen different forms of corals, right? Branching corals and mounding corals. So branching obviously have branches, and mounding corals look like big boulders. Some of our charismatic corals that are around here are the Acropora cervicornis, or the staghorn corals, as well as the elkhorn corals. And these have been responsible for as the major reef builders, especially in the Caribbean, because they create the habitat that fish really rely on. Mounding corals are some of the examples of the different types of corals that we have on our reefs. And corals reproduce in several different ways. There's sexual reproduction, where corals produce gametes, like these little dots in this photo, as well as asexual reproduction through fragmentation. So if a piece of a coral falls off, yeah. Just you wait, I got really into animation on this. It grows into another coral wherever it lands. And this fragmentation method is how you end up with some of these densely populated areas on reefs. And so here's we all know also that reefs are in trouble. And so what are some of the threats to reefs today? Overfishing, I agree. Disease, temperature, chemicals, nutrient pollution, ocean acidification, and other aspects of climate change. Development. There are so many different stressors that are affecting reefs in our backyard as well as globally. Plastic pollution. Corals are eating plastics. And it seems like a never-ending litany of what are harming reefs. And it's resulting in this shift often from corals that, as corals die, we see a shift towards algal reefs, which is making it harder to return to these coral environments. And so as corals are dying from these many different sources, one of the iconic ones is coral bleaching, which we've seen here in the Caymans. The Great Barrier Reef has been ravaged by it over the past couple years. And here's just an example of the start of a healthy reef, where we see that brown color that's indicating a healthy live coral. The expulsion of those zooxanthellae, where corals are dying here, there might still be tissue present, which would allow corals to engulf Symbiodinium and recover. But if that doesn't happen, we end up with dead reefs covered in algae. Another one of the major reasons for coral mortality is coral diseases. And coral diseases are really hard because some diseases look very similar to one another, and sometimes it looks pretty similar to bleaching or predation. Some diseases are easy to diagnose, like black band disease, where there's this characteristic black band. And it's also somewhat easy to determine what it's causing that kind of disease, because that black band is actually a cyanobacteria. And it's one of the few coral diseases where we know what the pathogenic agent is. But we also see white pox. We see the stony coral tissue loss disease, which is focused really on mounding corals and boulder corals and has decimated a lot of the reefs in the Florida Keys. And white band disease, which affects the staghorn corals. And from the 80s on, Caribbean-wide, we lost 95% of those populations. The problem with coral diseases in general, is they're really hard, or it's really hard to find out which pathogens are responsible for them. So one way in which we've tried to discover it is culturing bacteria from corals. Sometimes we find candidate pathogens, so we try and reinfect those corals, and sometimes we see similar symptoms, sometimes we don't. If we see similar symptoms, and then it's hard to get those bacteria that we expected to cause that disease back when we try to reculture them. And so this is a really active and kind of scary thing that happens throughout the Caribbean and other areas where there are coral reefs, where we don't totally understand what are the causative microbes between them. And in the white band disease in particular is hard because this disease wiped out many of the staghorn corals before we were able to really understand anything about it. So those are all kind of the sad things, but what are we doing about it, right? And one thing that we're doing is we're trying to restore coral reefs and boost their populations through coral nurseries, where the goal is to take some of these really important corals, grow them, and then put them back onto reefs. And so this is the for me, it's been wonderful working with CCMI because of their coral nursery. The focus has been on those staghorn corals, which are creating which create habitat that fish rely on. They're also they grow pretty quickly, so we can grow them to pretty large sizes pretty quickly to put them back onto reefs. And so, if anyone here is not familiar with nurseries, is anyone here familiar with nurseries? Okay, so we've got a scattering. Awesome. But if you're not, I just wanted to give you a primer on what it's like to create a coral nursery. So you go out, you find some coral populations. So here's a colony, and here's a colony. And you take a fragment from those colonies. So you take advantage of that fragmentation asexual reproduction. See, told you, I got into animation. And you hang them on something like this, which this is like a PVC frame. Well, it's not. It's just my PowerPoint skills. You allow the corals to grow in the nursery to a size that allows you then to outplant them on the reef, like so. Thank you. I appreciate that. So the common goals of people who have coral nurseries are to maximize growth and survival of the corals in the nursery, and to maximize the growth and survival of corals after they're outplanted, to boost coral populations. So how should we grow corals to set them up for success? So two things that I think about, or that I've been thinking about over the past year or so I've been working with CCMI, are the different types of coral growth and this kind of issue with disease. And I've been working in Little Cayman on the north side with their coral nursery. And so if we were to just dive right in, we would see the nursery, which is set up with these PVC frames with the corals growing on them like so. And it's an ocean-based nursery. There are different types of nurseries, but in this case, the corals are exposed to what are pretty ideal conditions. So they're able to experience the water flow that's in the ocean, or natural water flow and natural environmental conditions without any potential competitors that could harm the corals as they're growing. So the corals originally came from different colonies around the island. And so here's just a couple examples. You can think of these different cartoon images as different coral colonies. One of the things that we know is that this means they also have different genotypes. And so they're all the same species, right? They just have different, like you and your neighbor are the same species, but you have different genotypes. So there might be something about the areas where they're from, where they're located, that allowed them to locally adapt to those conditions, and that's why we see different differences among them. So there's more than just asexual reproduction going on. There's sexual reproduction as well around the island. So we take these multiple genotypes. These multiple genotype corals are we have 14 that are growing in the nursery in this sandy groove about 60 feet down. So to address different types of growth, first we have to think about, well, how do corals grow? They can grow longer, and so we use a total linear extension measurement by measuring all of the lengths of the coral's branches. They can grow heavier, which I think is pretty self-explanatory, or they can grow denser. And so this is a way that I like to think about this is a skeleton of the coral is a little bit holey. But how much skeletal material versus how much holes can change. The corals can change that aspect. So if the holes, that porous aspect of the skeleton is larger, you would expect to see a less dense skeleton than when the holes are smaller. You can think of it as like holy Swiss cheese. So if there's a lot of holes in that Swiss cheese, it's kind of flimsy. If there's not that many holes in that Swiss cheese, it's pretty rigid. Make sense? Cool, glad you're all here. Okay, so do corals always grow the same way? No. Yes, I agree. And are there trade-offs in growth? Yes, probably. If we're maximizing length, which is one of the goals of nurseries, are we doing that at the result of minimizing another aspect of growth, like density? So what in a nursery can affect coral growth? What about stabilization? So corals are growing in the nursery, they're hanging, they're able to grow in really any direction. But when they go out on the reef, they're stabilized. They're stuck on the reef and they can only grow up. So here's just an example of how these corals, when they're growing on the frames, they're really kind of, there's no one direction they grow, right? So they move with the current, which allows them also to, I'm having way too much fun with this, you're right. Which also allows them to eat more, maybe, which is allowing them to grow longer, potentially. Whereas corals that are stabilized, mimicking what conditions might be on the reef, as water moves past them, they're not going to move. So the trade-offs I expected to see are thicker, smaller corals versus thinner, longer corals, depending on whether a coral is stabilized or not. So I set up an experiment with the help of some of the trusty colleagues at CCMI in Florida, where some corals were left hanging, mimicking the conditions in the nursery, and some corals were fixed, mimicking the conditions as we would expect them, what we're going to do eventually on the reef. And so just as a close-up, here's a coral that's in the fixed position, and here's a coral that's in the hanging position when we first set this experiment up. And we looked at 3 different types of growth, so that longer measurement, how heavy the coral gets, a dry mass measurement, and then a density measurement, so how kind of thick and buff the coral gets. So I'm going to show you a couple graphs where the measurement type is going to be here on the y-axis, and the treatment fixed versus hanging. So this would be stabilized versus not stabilized. And for length, so growing just larger, we see that the hanging corals grow longer than the fixed corals. And we kind of expected this, right? Like, my hypotheses were pretty spot on. Yes, I just... So for mass, we see a really similar pattern. So now we've got mass on the y-axis. Corals that are hanging are heavier than corals that are fixed. Well, that makes sense. They're longer. So even when this is corrected for initial mass and for size, we still see this pattern, though, so that's pretty cool. For density, though, we see the opposite. So corals that are fixed are denser than corals that are hanging. So that's pretty cool. So if we look at this all together, corals that are hanging are longer, are heavier, but less dense than corals that are fixed. And the differences are obvious. So if you were to just look at corals in these 2 different treatments, you'd really be able to tell without looking at the silly graphs which treatment they were from and really what that pattern is. So this hanging coral, you can see is longer, has these skinny little branches, and this fixed coral is kind of stubby and meaty. So what might this mean when we outplant them on the reef? Hanging corals, they're larger and they're skinnier. That might mean they might break more easily. Fixed corals are smaller, they're stubbier. They might be able to withstand higher flow better. So if we think of during hurricanes, they're maybe better able to withstanding their position after they've been outplanted. So what happens to these corals after outplanting, though, we don't know, but we'll find out next week, I hope. So these corals, the next step of this experiment was taking them from their different orientations and putting them on the reef. So we did that in November, and I'm going to Little Cayman tomorrow to find out whether growing hanging versus growing fixed influences their growth and survival in that next phase. So if I do this again in a year, I might have an answer for you. So yes, the types of growth change depending on how we stabilize corals in the nursery. And so I'm going to switch gears now and talk about disease. And so as I expanded on before, disease is one of the critical issues in coral reefs right now, leading to coral death, but we have a lot of trouble understanding coral disease. And coral disease, especially for staghorn corals, because we don't we didn't have the knowledge or the tools at the time of the outbreaks that have decimated the populations, we really don't know that much about the disease ecology of, for the corals. And unfortunately, some of the corals in the nursery showed signs of disease. So here's an example of this signature of disease where you could see some of the tissue sloughing off of the staghorn coral. So we noticed this disease in May and monitored the corals from June until late October, early November, collecting microbial samples and also visually identifying the different health statuses of the corals in the nursery. So whether the corals were healthy, where there was no visible signs of disease at all, if there's signs of disease, so partial dead areas of the coral and active disease lesions where the tissue is actively falling off, and then completely dead, so there's no sign of living tissue at all. So I'm going to show you this graph. That's a GIF. I had a really fun time with animations. Where the proportion of corals is going to be on the y-axis. Time is on the x, but this in the corner here, the date is going to be changing for each as the graph grows. And the colors here indicate their different health categories. So black is healthy, orange is disease, and gray, sorry, I'm going to redo that. Black is dead, orange is healthy, and gray is diseased. So it's going to be the different types of bars are colored according to those health categories. So over the course of time, you can see that those bars are changing. And if we were to stop that, over the time course of the disease, we see that there was an obvious peak, and that happened in July. So what is maybe you noticed is that the healthy increased over here. We saw signs of recovery after that peak of disease. And so some corals, once the disease had run through, had started to regrow. We also saw some death, but we really lost less than 25% of the nursery. So this is the proportion of all of the corals in the nursery. But we have corals on multiple frames. And sometimes frames had lots of diseased corals on them, like you can see here. And sometimes frames did not have that many diseased corals on them. And so this plot that I'm going to show you, this is just one of the frames. So I'm going to show you for all of the frames. But it's the same colors as before. So that gray is the disease, the orange is the healthy, and the black is the dead. So across the nursery, this is how the disease moved through. This is over time. This is how the disease moved over time on every frame. So that's about 30 to 50 corals per frame. 30 to 15 to 50 corals per frame. And there's some patterns that jump out at me, and I don't know if they jump out at you, but I'm going to tell you what jumps out at me. So there are some frames that show that same pattern as we saw with the overall disease. So there's that peak around July, or there's quite a bit of disease on those frames. But then there are some frames where they stayed pretty healthy the whole time. Maybe there's a little bit of disease, but not that much. So what are the differences between these frames where there is disease or where there's not that much disease? Well, something that I haven't told you yet was that we know that there are different genotypes in the nursery, but we also have different arrangements of them. Some of the corals are arranged on frames with mixed genotypes, and some of the corals are arranged on frames where it's all the same genotype. So the colors here indicate what genotype they're part of. So for this graph, I divided the data. This is just looking at the proportion of diseased corals. The black is the single genotype, and the blue is the mixed genotype. And this is over time again. And just watch those dots move. So if you're a coral on a single genotype frame, the disease moves through those frames. But if you're a coral on a mixed genotype frame, maybe you got the disease, maybe you didn't get the disease. And that's pretty cool. So there's genotypic diversity on the frames increases resistance to disease. So that question that you asked earlier, what can we do about it? Well, one of the things we can do, at least in the nursery, is arrange our corals so they're next to other genotypes.
But in the real world, you can't do that.
But in the real world, it's a lot tougher. I think that's a conversation that we can start having, I think, after. That's much harder to answer.
Was the disease specific to particular genotypes, or was it the diversity that was preventing?
So it was not specific to specific genotypes. So even if I were to break this graph up by genotype, this pattern would still hold. So a coral that was from the same genotype but on a mixed genotype frame still showed on the single genotype frame this pattern, and on the mixed genotype frame this pattern.
This is for all the genotypes that you studied.
This graph right here is, yeah, this is across all genotypes. So that was pretty cool. But what about the microbes? So who might be causing this disease that we're seeing? So to look at this, we sampled healthy corals, the healthy area on a diseased coral, which we'll call apparently healthy, and a diseased coral. So at that interface in between the healthy and the diseased lesion. To do this, we take syringes and we agitate the coral surface a little bit and we suck up the mucus. And then we analyze the bacterial communities in the lab using genetics. And just to paint you a picture, there are about 70 coral species in the Caribbean. But on a single coral, there could be almost 2,000 different types of bacteria. So the diversity on a coral can be huge. So I'm going to show you a couple of groups or a couple plots where we're going to be looking at a single coral, which is represented by a bar. And the different colors within the bar are the different microbial groups associated with that coral. So those are the microbes on the coral. The size of that bar indicates how abundant that bacteria is relative to the other bacteria on that coral. So I forgot to mention this, but in 2017, one of our collaborators looked at healthy corals in the nursery. So I'm going to compare what we saw to what they saw when there was no incidence of disease at all. And so each of these lines indicate a different coral, and because this was a different project, they had 70 samples. What jumps out is this brown color, and that's all one type of bacteria that we're thinking is a symbiont of staghorn corals. So its presence is an indicator that this coral is healthy. So now let's look at the corals that are healthy but in the nursery during the time of the disease, so in 2019, and then on apparently healthy tissue but on a diseased coral. So here's the 2017 healthy coral, and in 2019 there are fewer samples because we were doing a more targeted approach. And what jumps out at me, and I hope at you, is we still see a lot of this brown group. So there's this stable, healthy community. So even on a diseased coral, you see this bacteria that indicates that the coral that's our indicator of coral health, as of a coral being healthy. So I'm going to show you the bacterial groups on a coral that's diseased. So what jumps out at me is that there are a lot more colors. So there's more diversity of different groups and a loss of that symbiont. And for corals, this is a really typical pattern that you see for disease, where there's an increase in the number of bacterial groups. Usually when you hear diversity, you think diversity, more diversity is good, which in a lot of cases that's true. But for corals, if you think of the coral microbial relationship as tightly coupled, the breakdown of that relationship is what might be resulting in this increase of different types of groups. So in this case, seeing more groups might be what's leading to the negative effects on the coral. So I wanted to know a little bit more about what microbes are associated with disease versus healthy. And so this is looking at a more specific level at the genus. This is that brown, what we're calling a symbiont. So it's really abundant in the healthy corals, and it's decreased its abundance in the diseased corals. And these 7 bacterial genera are increased in disease compared to healthy. One of those groups is Vibrio. And so Vibrio has been associated with, Vibrio cholera is associated with human diseases, cholera. But Vibrio has also been associated with coral diseases, including with white band here in the Caribbean. These other groups have also been associated with disease. And so these might be some of our major players that are either causing or working together to either cause the disease or as a byproduct of the disease. So some of the take-home points from this are that healthy-looking tissue has healthy-looking microbes, which I think is really cool, in part because we were able to recognize that even on a diseased coral, there are healthy aspects to it. So it's not completely, it's not fated to die, potentially. And knowing what that healthy microbial community can be really important when trying to forecast or predict over time. And that disease might not be caused by one microbe, but by many microbes working together, as we can see that there's not just one that's increasing in the presence of disease, but 7. So, in the nursery, we've learned stabilization changes growth, that growing corals on mixed genotype frames decreases the potential for disease, and that corals that, even if corals are sick, they have healthy microbiomes associated with them, and there's a pretty stable, healthy microbiome community or bacterial community for staghorn corals. So this is what we've been doing in the nursery over the past year, but we're also up to some other projects that are more in the development stage. So currently, we have an outplanting project where we're asking, can a coral go home? So I alluded to this earlier, where we have those colonies around the island where corals originally came from, and they have different genotypes. Those differences might also lead to different traits of those corals. But corals have been growing in the nursery for 7 years, so maybe those traits are really flexible, or maybe they're adapted to those original conditions. So we're asking if corals perform best in their home locations, if corals can be flexible enough, so change these different types of growth or survival strategies to be successful in new and different environments, and which of the genotypes that we're outplanting perform the best overall. So which genotypes grow best no matter where we outplant them. And for this, we've outplanted these corals to 4 sites around Little Cayman that we're also checking in the next 2 weeks, so stay tuned. The other thing I wanted to mention before I leave you is that there are other diseases to look out for that are not in nurseries but are in the wild. Have you heard of stony coral tissue loss disease? Yes. So this is just a timeline of how it affected the keys. It can move really quickly, leading to 60% coral death within even just a month. It can follow a bleaching event, and I know we just had a bleaching event here in the Caymans. It's really hard to determine whether or not stony coral tissue loss is present because it can move so quickly, and by the time you realize it's happening, it's gone. And so what I'm going to implore for all of you to do as you're diving, as you're exploring the reefs, is to keep an eye out for stressed corals that are showing signs of tissue loss at, or like bright white skeleton, the tissue sloughing off at the interface between the healthy area and the diseased area. If you see something that looks like this, even if you're not sure, please let me know. And with a photo or a description of the site, the date that you took it, and you can send it to my email. I really appreciate your attention, and if you have questions, I can answer them now. Yeah. I think I saw you, then you, then you.
Have you got a hypothesis as to why diversity should be protected?
Yeah, so one of my hypotheses is that there's, so if a coral is the same genotype as all of the corals around it, then if there's any protective gene or metabolite that could be produced by a coral of a different genotype, that wouldn't happen if everything is the same. But if there's some genetic difference between coral A and coral B, like genotype A and genotype B, then a coral, once it's touching another one, wouldn't be able to spread the disease past that resistant genotype. So something that we would need to really delve in further is which genotypes might show resistance compared to others, is if there's specific ones.
But I was thinking,
Yeah.
as in human medicine,
Mm-hmm.
when a bacterium is resistant,
Yeah.
then you hit it with a combination of antibiotics
Mm-hmm.
and overcome the resistance that way. So maybe each different genotype contributes a metabolite, the combination of which is toxic to the bacteria,
Oh, that's cool. That's a really great idea. Yeah.
rather than a single metabolite.
No,
a combination.
yeah, the combination. I think that's a really good idea. I think that this pattern is a really ripe ground for generating more hypotheses and questions. It's really, yeah, I'm pretty excited to move forward. I really appreciate that. There was somebody over here. Yes. And then you. Yeah.
Have you ever, and going back to kind of the microbiome of the corals, so you have identified like symbiotic bacterium species?
So that's our hypothesis, yeah.
And like isolated them?
Nope. So we're at the, so what's cool about this guy that we've identified as a potential symbiont is its genome was recently mapped, genotyped. We have the entire genome now. And so we're trying to figure out ways that we, to at least know what the genome of this specific bacteria is, and if there's any potential methods for culturing them.
That's
The issue,
orange microbe?
yes,
Okay.
that orangey brown
Orange
one.
brown.
Yes. So like one of the difficulties of the coral bacterial communities is it's really hard to culture a lot of the bacteria that's associated with that. But there's, yeah, but as a scientific community, I think we're trying to become a little bit more creative on how to do something like that. Yeah.
So, well, I mean, from kind of the graphs that you've shown, it looks like the bacterial species and kind of outcompeting the other kind of disease-causing bacteria. So I was wondering if you would try or will try any kind of experiments to maybe transplant that bacterium to disease corals to see if that can have
Yeah, I think that's a great idea. That would be a great next step. Mm-hmm.
the corals in the wild that actually might be able a way to kind of combat that and not just in the nursery.
Yeah, that was a great idea. So what was just suggested was if we could find a way to culture, take some of that bacteria and use it as a probiotic, I think is what you're trying to get at, right, on corals that are diseased. And that's something that for the stony coral tissue loss disease, they're trying to do in the Keys right now. Yeah, like hopefully something like that will work. It's just really hard to scale some of these ideas up. Question?
I wonder, has any research been done in coral gardens or coral, there's a lot of coral, the coral being exposed to a lot of traffic from motorized boats as compared to coral being not exposed to that type of traffic, but just to, let's say, sailing, where you don't have all of the various things that come out of it.
Yeah, no, I think that's a really good question. So this kind of comparison of, like, can I call it like a highly disturbed or heavily disturbed area where there's boat traffic and anchoring and motorboats versus somewhere where there's a lot less use. And there has been a bit of that kind of comparison that's done, maybe not as specific, but like heavily populated versus low populated areas, and looking at coral cover comparisons. And those less disturbed areas tend to show higher coral survival and recovery more so than the more heavily disturbed areas.
So can we go back to sailing?
Sounds great.
Get rid of the catch.
Yes.
So in the first half of the presentation, you were talking about the different structures of the coral.
Mhm.
So it seemed that you were indicating that the squat coral might be more resistant to wave damage.
Mhm.
Is there any, well,
Sorry, go ahead. Keep
anyway,
going.
try to figure it out.
Yeah.
But is there any potential kind of benefit of having the kind of thinner skeleton, maybe it's more flexible in the waves? Is that a potential benefit to the microbial
It's been used for a million years, so it's.
promoting?
So there's possible that maybe it wouldn't be more flexible, but maybe it would break off. Maybe that fragmentation, though, can help reseed other areas of the reef. So there is still some question about, right, like what does it mean even though they have these different growth characteristics? Is it where we plant corals that will affect their success after outplanting for these different types of morphologies, which is just a whole ripe area for,
Yeah. it seems like
Yeah.
it's too early, maybe right
but that's a
one
great,
side or the other.
but it's a really great point, is that whether one is better than the other is really hard to say at this point. It's just more that there's differences in trying to maximize growth and boost populations. Maybe knowing what these differences are early influences what their success will be later. Yes.
Back to the potentially symbiotic
Mhm.
microbe that's for the elk.
For the
and
staghorn,
coral,
yeah.
It's the staghorn. Is that applicable to other corals as well, to the staghorn?
So that's a good question. I've found this same bacteria in elkhorn and in staghorn, but it's not the same potential symbiont as in a Porites coral, for example. Some of like the mustard corals around, those have a different candidate symbiont.
And would potentially, when you're looking at these various microbes, if you went into different corals, that perhaps one might be helpful for one and harmful for another?
That is possible, right? Because they're potentially co-adapted to
Corals
that
and
environment.
everything
Yeah.
else, yeah.
And so that's,
Sprinkle, you can end up with a microbe.
yeah, right. It's
Yeah.
we still have a lot of work to do, I think. Question?
I worked in Acapulco, Mexico last summer.
Yeah.
And we saw a lot of the stony coral tissue disease, like within the span of the summer before. And then that summer, I think they estimated that in like the Bay Area, they lost half of their corals,
Oh,
or
wow.
their stony corals. But I was wondering, because like you said, we saw it spread really fast, if there's any way of like fragmenting the corals or removing like the infected tissue to protect like the healthy tissue. I don't know.
That's a really good question. So the Florida Keys has in the Florida Keys where there's been a lot of stony coral tissue loss, one of their mitigation strategies has been trying to remove some of the healthy corals from those areas and removing the disease portions and trying to rehabilitate some of the corals that did show disease. And they haven't published anything about that yet, but that I know of. But I did visit the Florida Aquarium recently, and it seemed as though those ones that did show disease, they're still in quarantine, and so we don't know. But it might be a down the road strategy that we could think about using. But again, it's a hard one to do on large scales. Yes.
Just a question about your nursery with the hard structure versus the dangling.
Mm-hmm.
One of the discussions we're having here with our coral
Yeah.
nurseries is about the concept of leaving trees as gamete producing seed trees versus outplanting.
Yeah.
Have you noticed any difference in the gamete production between the hard attached versus dangling structure?
So we didn't look at that for for those corals. Dagny, did you, by any chance, you guys didn't tend to any of those? No. So I'm not sure, but we did look at some of the gamete production, at least by the disease corals, and they were still producing gametes, which was pretty cool. But that's a great question, and that would be a really nice follow-up to what we did. Yep.
Going back to the floating versus sessile corals, in your first picture on the actual anatomy of the coral, there's the layer of living tissue and mineral inside. So when you say more dense, is there more mineral deposition that's happening in the interior of the coral?
Yeah, that's my hypothesis. So I'm hoping, so one of the measurements that I haven't shown here, but might be able to get at that without like sectioning the coral and looking at it through like a CAT scan or something like that, is trying to plug up all those holes using wax. And so that'll more definitely get at that question.
The other question I had was again on the genotype.
Mm-hmm.
Is it an assumption that corals from different parts, coral collected, harvested from different sites, have a different genotype, or have we actually looked at the entire sequence and seen differences?
Yeah, that's a great question. So for 5 of the corals, we definitely have the sequences, and for the remaining ones, we are hypothesizing that they're different because they come from different colonies, and the colonies that they come from are pretty isolated from one another. But that's that assumption hopefully will be tested in the near future. Yep.
Is it the ultimate goal to take these corals and replant them out back in the wilds?
Yeah, that's one of the goals.
From Little Cayman, just on Little Cayman, you wouldn't bring them to Grand Cayman?
So that, yeah, there's nursery efforts on Grand Cayman and on Cayman Brac where it's been pretty like local, where we outplant on.
The question is, nothing's being planted out. I mean, when you go diving, you don't see, anything that you grew now planted and growing.
Not yet.
Yes, yeah, we do. So we do have outplant sites around Grand Cayman. There's several of them where we have multiple different genotypes planted on the same coral head. So different coral heads have been selected. At the moment, like I said, there's a discussion of what type of efforts are going to go towards outplanting versus expanding the nursery program as gamete seed trees.
Did you say that there's only 70 kinds of coral in all of the Caribbean?
70 different species, I do believe,
In the entire Caribbean?
in the Caribbean, but that's not globally.
No, globally, there's thousands in the Pacific.
Yeah, just
So
the Caribbean
other
is a
question
much.
would be, does it pay to take coral from the Pacific, different coral that you don't have here, and bring it here?
That's like so many years of evolution that would make it really difficult for that to.
It's like anything. With
So
coral.
yeah. So I mean, like there's some. I think. Yeah. So there are a lot of differences between. I mean, beyond just like evolutionary, they're very different between them. They're different species. So they look really similar, but they're not the same species in the Caribbean versus in the Pacific. I think that that's a pretty radical kind of like, if last-ditch effort, future, hopefully like way down the road, maybe solution. But for the moment, trying to think about what corals are able to survive already in these areas and trying to propagate those.
But didn't we have a speaker who suggested that because the sea temperatures were rising,
Oh, I'm sure.
that there was some coral that did better in warmer seas and that might
Right.
work better locally?
Yeah, she said that.
Yeah, I don't know where she was thinking of transplanting it from.
So
What's
that.
happening there, moving it,
Was that you?
the sea levels were warm.
Well, luckily that speaker is here tonight, so if she wants to address that question, she absolutely can.
I was discussing in terms of the deep reef, so because the reef down below 100 feet is significantly cooler than the shallow water reefs, they seem to experience less outbreaks of disease and bleaching. And so there is a possible hypothesis that we could, for example, set up nurseries down in deeper waters where it's cooler, where they're less likely to expose diseases, maybe more resilient. And then, as the disease passes by, then we could maybe move them back up to the shallows, that kind of idea. There's also along that same line, you could assist northerly migration with corals, so assist them to move up the coast of the United States or to more northern reef locations in the wider Western Atlantic.
question.
You're
But
finding
still
that,
using land phase-in.
Yeah.
you're finding that mixed genotypes were doing better than single genotypes. Made me think of this question: what about mixing different species?
That's cool.
Would they do better than a single species?
Yeah, I mean, I think that I would have the same hypothesis that yes, they would probably do better than a single species. It would be even harder for a disease to jump across species than across genotypes would be my expectation.
Or even have some kind of protective function for the species that would otherwise get sick.
Yeah, there's actually a really interesting study that came out rather recently where mixing different types of corals can also boost coral growth, which is found also in like mixing agricultural groups, right, where it boosts growth because there's different trade-offs because of competition. There's another question in this area.
I was just going to ask, is it just staghorn coral that you're doing the research on, or are you doing any other type?
That's a great question. Right now, just staghorn coral. There's some elkhorn coral that's been growing, but we haven't been
So it's the
researching
branching
that
corals that you're dealing with.
right now. Mm-hmm. Yeah.
Does the sargassum weed affect the staghorn pollen?
So sargassum, when it's like floating or like when it's attached, it can affect the staghorn. When it's floating, it can shade them. When it's attached, it could like compete with them either by hitting them or overgrowing them. But in the nursery, the staghorn or the sargassum hasn't been as much of an issue because it's 60 feet and it's generally there's just generally less light, and so it's also in an area where there hasn't been dense mats of sargassum that just remain there.
I was just wondering about the microbes attached to it.
Yeah, that's a great question. So the sargassum and other types of algae can affect the microbial community just in the reef waters. And so if there's a lot of sargassum and in an area where the water is not moving very much, that could be one of the factors that could really, really harm corals. That's something else I'm actually really interested in. So,
I know there was a paper a couple years ago, I think it was in Southeast Asia, that showed corals that had come in contact with plastic had like up to 20 times higher incidence rate of disease.
oh yeah.
Is that something, that's been looked at in the Caribbean, or, whether areas with more plastic pollution are having higher rates
Of disease
or faster spread of disease because it's carrying microbes
from place to
reef
place.
reef,
Yeah, that
skeletal
makes a lot of
plastic.
sense. I don't know anything off the top of my head, but that's a really good thing to keep in mind.
And on your frames that had multiple genotypes, did they all have the same number of different genotypes? Or there's some that say 2, 3, 4, and then was there any correlation between
Yes.
recovery speed if there was more?
That's a great question. They did have different numbers, and I haven't looked at that yet because this is
Just to
That's a
back to the
great-
first question,
yeah,
if
and
you were to
that's
actually gauge it, if there's
one
more
of
there,
the
there's correlation,
yeah, that's one of the exciting things. That's like, really delving into the data is what I'm looking forward to doing right after this field season. So this has been really wonderful. I appreciate you all listening, and I appreciate all these questions. And again, if you have more, or if you think about something later, I would be happy to answer questions via email or tweet me. I'm trying to get into Twitter. But I think that's the end. So thanks.